So in this video we are going to cover these topics, that is introduction to micromeritics, definition of micromeritics, importance in pharmacy, applications of micromeritics in drug formulation, and particle size distribution, methods of particle size analysis, particle shape, surface area, porosity, density of powders, flow properties of powders. What is micromeritics? That is definition of micromeritics. So here micromeritics is the science and technology which actually involves the study of small particles. The study mainly focus on study of their particle size, particle shape, study of surface area of the particle, study of porosity of the particle, and study of density of the particle, and study of flow properties of the particles. Now the importance of micromeritics in pharmacy. So actually it is going to affect the drug dissolution, drug absorption, and it influences the powder flow properties during tablet manufacturing. And micromeritic study also helps in achieving uniform mixing of the ingredients during the formulation process, and it determines the stability and quality of the pharmaceutical products. So because here we are studying the actually the physical characteristics of the powders, of powder particles, like size, shape, porosity, diameter, all those things, it is going to, these are the physical properties which are going to help us mainly in the formulation to enhance the drug dissolution, drug absorption, and to achieve the uniform dosing, uniform mixing of the ingredients to obtain the uniform dosage form. And it also helps in maintaining the stability and quality of the pharmaceutical products. The first property is particle size. Particle size, it is nothing but a linear dimension or diameter of a particle, which is actually expressed in suitable appropriate units like micrometer. Suppose here it is a particle. So now what is the size of particle means, or what is the meaning of size of the particle means? It is a linear dimension. Linear means straight line, straight line dimension, or a diameter, of a particle. So it is the important parameter which is going to affect the physical as well as pharmaceutical properties of the powders. In the simple definition, we can say powder size refers to, or particle size refers to how large or how small the particle is. Here there are some common size ranges like coarse particles. The range is below 1000 micrometer. Fine particles, it is in between 10 to 1000 micrometers. Ultra fine particles, it is up to 10 micrometer. What is particle size distribution? So it is the distribution of particles of different sizes which is present in the same powder sample. For example, here we are taking a powder sample which is having different sizes of particles. Now the particle size distribution means it is in the powder sample. Here we can see there are different sizes of the particles. Some are circular shape, some are oval shape, oval shaped particles are there. They are distributed in the powder sample. and it is expressed as percentage or fraction of particles within the specified size ranges. In the simple definition, we can say particle size distribution shows how many particles of each size are present in the same sample. Now, how can we analyze the particle size determination? How can we find out? So here there are different methods by which we can determine the particle size distribution: by the help of sieve analysis, by the help of microscopy, by the help of sedimentation method, and by laser diffraction method. Now the another property that is called as surface area. So surface area is the total area which is exposed on the surface of a particle. In the simple definition, we can say surface area refers to the amount of the exposed surface on the particle. For example, it is a powder particle, then this is called as the total area which is exposed, that is exposed surface area. So what is the importance in pharmacy? It mainly affects the drug dissolution rate. Means the powder particle, the surface area which is exposed, the surface which is exposed or which is exposed to the water, so which comes in contact with the water, it mainly determines the dissolution of the powder particle. The dissolution of course influences the drug absorption and bioavailability of the drug particle. Then it affects the adsorption of moisture and gases and influences the chemical reactivity as well as stability. Main logic is smaller the particles size, larger the surface area, and faster the dissolution. So if you want to achieve dissolution rate faster, then if we decrease the particle size, that will offers larger surface area, that definitely increases the dissolution rate. Types of surface area: first one is absolute surface area, another one is specific surface area. So absolute surface area means total surface area of all particles, means the number of particles which are present, total number of particles which are present in the powder sample, altogether surface area is called as absolute surface area. Another one is specific surface area, means this refers to surface area per unit weight or volume of the powder. Methods of determination: surface area is determined by adsorption method and air permeability method. And the next property is porosity. So porosity is the percentage of ovoid spaces. Here ovoid space means empty space which is present in the powder bed or compact relative to its total volume. Simple definition: porosity indicates how much empty space is present or exists between the particles in a powder mass. Suppose it is a powder mass. So this empty space is called as porosity. Porosity, that is ovoid spaces. Now here is the formula to calculate the porosity. Porosity is equal to VB minus V rho divided by VB into 100. So here VB means bulk volume of the powder, V rho means true volume occupied by the particles. So here bulk volume and true volume we will discuss in the next slide or upcoming slide. Then now the importance of porosity in the manufacturing of pharmaceuticals or pharma products. So it is going to affect the powder flow properties. It influences the tablet compression, It affects the penetration of the liquid into powders. It influences dissolution and drug release. Here it is important in granulation and capsule filling. Higher the porosity, more empty spaces or void spaces between the particles. So here porosity is going to affect the packing and compaction behavior of the powders. The porosity is defined as it is the ratio or percentage of empty space or void space which is present in a powder bed to the total bulk volume of the powder. Means here porosity is equal to, it is the ratio of percentage of void spaces or empty spaces, that is space between the powder particles, divided by total volume or bulk volume of the powder sample. Now the another property, that is density. What is density here? So density means it is the mass of a substance per unit volume. Means the density is represented by a symbol called as rho is equal to mass divided by volume. Here rho means density, m means mass, v is equal to volume. So this density is of different types. The first one is true density. True density means the mass of the particles divided by their true volume excluding pores and void spaces. So here true density means, for example, here are the powder particles. Suppose here are the powder particles. Now the true density means here we are going to leave this empty space, that is void space, we are not counting, and the small gaps which are on the surface of the powder particles, they are called as pores. Small gaps or small holes which are present on the surface of the powder particles, which are called as pores. This also we are not going to take. We are leaving this pores also and the empty space also. After that we are going to count the remaining things in the true density. Means it is the actual or original density of the powder particles excluding pores and empty spaces or void spaces. Now the bulk density means what? Bulk density means it is the mass of a powder divided by its bulk volume, includes spaces between the particles also. So as we have discussed the earlier slide, true density means we are going to leave the space between the powder particles and the pores or the gaps which is present on the surface of the powder particles, that we are going to leave in the true densities. But here in the bulk density, so we are going to consider each thing, that is, for example, it is the powder particle, and here we are going to take the weight or size of the powder particle as well as the empty space of the powder particle and the pores which are present on the surface of the powder particle. All these we are going to take under consideration, or we are going to add all these things for the bulk density. Bulk density means the total volume, that is, it includes the void spaces, empty spaces between the particles as well as the pores also. Now what is tapped density? So here tapped density means, for example, we are taking the powder particles in a measuring cylinder. So initially the powder particles are present in the here up to this mark. Then we are going to tap this measuring cylinder for a certain number or certain times. We are going to tap this measuring cylinder which is containing the powder particle sample. After tapping. After tapping, what happens now? The volume of the powder particle is going to reduce. As compared to this. This type of density, that is the powder volume which is obtained, or the density which is obtained after mechanically tapping in a measuring cylinder, it is called as tapped density. Here Y means the volume decreases, the powder particles settle, and it will reduce the volume, and it will reduce the empty space between the powder particles. Now, importance in pharmacy. So these properties of the powder particles helps in designing the dosage form, affects the powder flow properties, and important in tablet compression, and useful in capsule filling and packaging. So here is some GPAT like questions based on this topic. Like first question is, Micromeritics is the science and technology of option A drug metabolism, option B small particles, option C drug formulation only, option D pharmaceutical analysis. Answer is B, that is small particles. Now the question number 2, that is, which of the following is not a parameter studied in micromeritics? Option A particle size, option B particle shape, option C surface area, option D molecular weight. The answer is D, molecular weight. Which method is commonly used for particle size analysis of a coarse powder? Option A electron microscopy, option B sieve analysis, option C X-ray diffraction, option D spectroscopy. The answer is option B, that is sieve analysis.